Our first speaker today is Sabine Langie from the Maastricht University, and she's going to tell us more about DNA repair, as Amaya mentioned yesterday.
Thanks, Marco. Okay, so talking about Comet, you've seen from Amaya that you can look at DNA damage with it, different types of DNA damage. But there's other biomarkers of disease that you can actually study. So monitoring environment, okay, at DNA damage, what is the exposure? But also antioxidant status, as well as DNA repair. And DNA repair is the one I'm going to focus on in this talk. Who remembers which types of DNA repair are there? So one is basic excision repair. Yeah, mismatch repair, thank you. Nucleotide excision repair, yes. Homologous recombination, yeah, it's one of the double strands DNA damages repair, yeah. The last one is maybe the hardest, the direct reversal. You can say, okay, it's not really maybe a repair, but still it is. It's just simple removal. So imagine you have alkylating agents which add a methyl group to your bases. Instead of cutting out the base, what the other ones here at base excision and nucleotide 2, they just cut off the group, the chemical group, and then it's reversed to the original base. So that's why it's called direct reversal. And then indeed, the mismatch repair has been mentioned. Basic excision repair, which mainly deals with the single strand breaks or like the damaged bases. So it cuts out damaged bases. Nucleotide excision repair cuts out bigger ones. So the bulky adducts, or if you have, for instance, UV dimers. And then indeed the double strand DNA repair, which is both homologous and non-homologous recombination. So you have two. Actually, for today, I'm only going to focus on two. This is just to refresh your mind again. I think it's very obvious why we have DNA repair as. So it's to prevent mutations from occurring and cancer or other aging processes to happen. So that's why we really need DNA repair to occur. And like I say, today I'm going to focus on two, namely basic excision repair. Again, I have a lot of enzymes called DNA glycosylases that cut out a base. Here's an oxidized base, but it could be alkylated and methylated. And then it replaces that base through the process. And then the nucleotide excision repair is the one that cuts out the bulky adducts, but it does that in replacing a series of nucleotides. And it's a more complex process because there's a lot of more enzymes and cofactors working together. I'm not going to go into detail on these processes, but just remember these two processes. Okay, then how do we measure DNA repair? And the most simple way that you might have maybe already come across is doing a Comet assay, like Amaya explained yesterday, but do it over time. So you expose, for instance, your cells, and then you're going to see, okay, how is the repair either accumulating? Usually in the beginning, it accumulates a bit, and then how is it removed over time? And then by the slope, you can get an estimate of how quick repair has occurred. But you can imagine this is quite time consuming. This is now 4 time points, but some people take even more time points. So it takes a lot more time to get to the data. So that's when Andrew Collins actually came up with the in vitro DNA repair assay, the Comet-based one. There's other in vitro DNA repair assays that are based on oligonucleotides or plasmids. But the one I'm going to explain to you today is based on the Comet assay. And well, Andrew set it up for basic excision repair with a compound that's called Rho, 198022, but we always say Rho, plus light because that induces singlet oxygen and other oxidized lesions. And recently we used potassium bromate, and we're using it more and more now. The other way is to induce UVC lesions, or you use a compound that induces bulky adducts. In this case, we set it up with PPDE. You can think of it as one of the chemicals in cigarette smoke. PPDE is benzoaparindiol epoxide. It's one of the metabolites. And it induces these bulky adducts, and then it's a substrate for nucleotide excision repair. So the exposure you choose here is important for making your substrate cells. So that's the main difference with the other Comet assay, that here you don't just expose to the compound of interest or the exposure of interest. You really select your exposure depending on if you want to study base excision repair related processes or nucleotide excision repair related processes. Whereas then your sample of interest is on the other side. Now I didn't draw the cells, but here tissues or blood or cells in culture, you can extract the proteins out of them. And in these protein extracts, there's also enzymes, of course, so the DNA repair enzymes. And then the idea is that you incubate these enzymes with your substrates. And I already talked about nucleoids. So after lysis, you get these nucleoid-like structures which contain the lesions, your substrates. And then the enzymes can go there and cut. And the rest is standard Comet assay procedure. This maybe makes it a bit more clear how the principle works. So imagine here is an oxidized guanine. And then you add on your extract. It will excise the guanine and incise the DNA. And that's actually where the reaction stops, because we don't add any nucleotides to the mixture. So we only have the process going until the incision step. And then because of the alkaline unwinding, your DNA will unwind there, because this incision you can compare with a single strand break. And then it will unwind and you get fragments or loops relaxing, and then you get your comet tail. So indeed, so in DNA damage Comet assays, a long tail means a lot of damage occurring, but here it's actually a lot of incisions occurring. So the more the enzymes incise the substrate, the longer the tail. So here it's a good thing. So the longer the tail, the more DNA repair activity has been taken place. So that's important in terms of interpreting the data. So how do we then calculate DNA repair activities? Maybe very practical, but just to give you an idea of all the controls you need to include. So always a background control, which is just incubated with a buffer. And these cells have been prepared just with PBS. They have not been exposed to your stressor, let's say. Then you have the ones that have been exposed, for instance, to UVC or this row compound, but are also incubated with buffers. So that's like your treatment control. Then you have the ones that have been non-exposed, but incubated with the extract. And this is just to see if it recognizes any background damage. Maybe there has been some natural background damage in these cells. And then the exposed one, this is the more specific sample, which is what recognizes everything. So it recognizes your endogenous damage as well as the substrate lesions that you have induced. So first you subtract the background from all your values, and then you're going to take the difference between the other ones. So you do 4 minus 3 minus 2, and that gives you the net DNA repair activity. So it's a bit more complex than calculating the net FPG sensitive sites that Amaya explained to you yesterday. So here you need to include a bit more controls. Is that clear for everyone? Yeah. Well, if you want to know more, there is this Nature Protocols, which you will see later also QR code for. Here we show 2 gels per slide. You can actually also use the 12 gels. Amaya showed you yesterday also. You can even top it up to 48, 96. But the 12 gels has the advantage that a device has been made that actually allows you to incubate in every well. So in between here, you might not see it very well, but there's a rubber gasket, we call it. And because of the pressure that you put on it, actually the wells are nicely isolated. So in every well, you could do a different incubation. So that increases your throughput a lot. So this is a paper I talked about. So it's one of the 3 Nature Protocol papers that we published to actually standardize the way people do the assays. So as I already said, these are the main differences. So maybe this gives you a different perspective. So on one side, you have to prepare your substrate cells. You can do this any day and just freeze them as aliquots. And then on the other side, you have your samples to make your protein extract. Also this you can do any day and freeze back in the -80. And then on the day of the experiment, when you have a lot of samples coming together, you can run them in the Comet assay. And the rest is standard Comet assay. So this is the QR code that I was talking about, but this is actually for the YouTube channel. So Amaya said she was not in a film, but if you check out these films, she and I have been very busy with creating films as part of the H-Comet workshop, or H-Comet project, cost project. So there's many of them. Here is the one on DNA repair, but there's also the one on DNA damage that Amaya explained yesterday, and there's other interesting videos if you're interested to start performing the Comet assay. Okay, so now everyone follows how the assay works? Yeah. Okay, how can we then use it to do biomonitoring and use it for other applications? Well, these are the fields that it has been applied to so far. I have to confess, it was mainly human studies. In the aging studies and nutrition, there's also been animal studies a lot, but a lot of the work focused on nutrition. So I'm first going to zoom in there a bit and then go back to the environmental biomonitoring. And as you might know, nutrition can modulate DNA repair in different ways, mainly via the antioxidant statuses, but also fatty acids and so forth. This might be a bit small, but the most important thing is that you can see the colors of the boxes. And yeah, I put the figures of which type of food components or nutrients have been used. You can see that there's a mixture of effects. The green ones resulted in an increase of DNA repair activity. The yellow ones did not really find a difference, and the red ones, which are luckily not that many, found even a decrease in DNA repair activity. Is that bad? Is it not bad? I will come back to that later, how to interpret this. One thing that we do fancy is this study by Andrew Collins. I actually repeated it a few times with the students, but apparently eating kiwis a day can increase your base excision repair activity. You might know that they're very rich in antioxidants, vitamin C especially, and 1 kiwi a day is already sufficient to increase your base excision repair activity. Then they tried to repeat it with the golden kiwis, and although it does reduce the DNA damage, you can see here before and after, it did not have an effect on the repair. So the 1 kiwi or the other kiwi is not the same, just to show you how different reactions can be. Then what's also important is to include genetics, because we all react different to food, right? And the interaction. So to give you one example, there was a study that I did in my PhD. So first we looked at genetics. This might be a bit complicated to understand, but imagine, I don't remember how many genes we did. I said it there, okay, 7 genes, and we studied 10 SNPs. And for every polymorphism, so for every mutation, we then calculated the allele score. So if you were heterozygous for the mutant allele, you get a score of 1. If you were homozygous for the mutant allele, you get a score of 2, and then you take the sum. And you can see if people carry more of these mutated alleles, they have a lower repair activity as we expected. But then what if we do look at the interaction? So we did an intervention study with blueberry apple juice for 4 weeks. And there was a washout period in the beginning. And then we studied before and after nucleotide excision repair. And we saw this, very disappointing, right? No effect of the intervention. But why was that? Then we looked at the interaction. This is just one example. There were other genes that correlated with it. But for instance, for this, ROD23B is involved in nucleotide excision repair. You can see that the ones carrying the double mutant allele for this gene, they actually do benefit from the intervention to increase their nucleotide excision repair activity, while the wild types did not. So just to show you that it might be important to see, like, are there susceptible groups that benefit more from an intervention or not? Okay, then going to human biomonitoring. It's mainly occupational studies that I will show today, but if you want to know more about it, there's another paper you can read where studies have been summarized. It's a bit older, but still a good overview. First study to show you in relation to pollution, and yeah, this is occupational exposure, styrene workers. You can see that indeed their basics in repair activity was, increased. Apart from the males, it was slightly decreased, but there's actually no statistical difference. But for the females, it was decreased due to the styrene exposure. The same was seen, and then smoking. So they also looked at smoking. Smoking, actually, if you just look at the controls for now, smokers had a higher DNA repair activity. Why could that be? Yeah, indeed. So that's again why I said in the beginning, like with the table of all the nutrition, like is it bad or good that we see a decrease? Indeed, it could also be that you see an increase in your repair because you get a trigger. You get exposed, or your body starts producing more repair enzymes to cope with the exposure. So that's probably what's happening here with the smokers. The same thing was seen for asbestos exposure. Yeah, there's all the damage markers, but to focus here, we see again that the exposed compared to control ones, actually the males had a slightly increased repair and the women had a reduced repair. So you also see it for asbestos, the same trend. And the smokers, we did not see a clear effect here. And again, when you talk about genetics, they saw that there was a lot of variation between the people, even within the group. So these were the exposed ones, and these were the non-exposed ones. And you can see there's a lot of variation there. So also they decided to look at the genetics behind it. And they found that there was an association between the GSTM1, one of the metabolizing enzymes, and XRCC1, which is involved in nucleotide excision repair. And you can see that, they show a significant correlation with the exposure. So they interact with the exposure. Also, their genetics is important to take into account.
Is it a crossover study, or was it two groups in parallel?
It was just two groups, two occupational groups that were studied. Yeah. There's another study from the same groups where they even looked at different plants around the city. And also there they could see differences between plants, but there was not a crossover. So coming back to what does it mean, a low repair or a high repair? I mean, if you see these data, so you measured it 4 times in this person, 4 times in that person, DNA repair, who is the more susceptible one according to this picture? Is it person 1 or 2? Who says person 1? The others still doubting? Come on, guys. Who says person 1 for the susceptible? 3 people. Okay, 4 people. Person 2? Doubting people. Maybe it's easier if you see it like this. So if you take into account the exposure, who's the more susceptible now? Who says still person 1? Maybe it's easier to do it like this. Who is the more susceptible now? What if we move this up? 1 or 2? Maybe a show of hands. Who says person 1 is the more susceptible one? Come on, guys, hands up. Person 2 then? 2 hands. It's person 1 in this case, because it has a low repair, but if it has a high exposure, then of course it's more at risk because the repair cannot keep up with it. Well, here, of course, it depends how long the body can keep up with a high exposure. So maybe after a while, repair might go down, but at that point, the body can still keep up with it. While at this point, it's a danger to the person. So there, it's the more susceptible one. And it may be different depending on the repair methods. So we've seen that bear and nare do not always react in the same way to nutrition or an exposure. So you really have to look at the different types. So this is just to show you whenever you design a study or set up a study, it's always good to look at different or multiple biomarkers in parallel so that you get a complete story. Okay, what about environmental biomonitoring, where the whole summer school is about, actually? Well, the sad news is that it's not done a lot with the repair assay in environmental biomonitoring, but that doesn't mean it can still be. So actually, we have done a lot of improvements over the years, and now that the Nature protocol is out there, but also very recent data from a bachelor study, we try to work with frozen whole blood. Why? You might have, no, we haven't said it yet, I think. Anyone talked about frozen blood in the comment? No. Huh? The next one. Okay, yeah, true. Ilou will show you more. But for the repair, so I'll not say everything, but Ilou will show you how you can use frozen whole blood. Why is this so interesting? Because you don't need to isolate white blood cells. It's a lot of processing steps. And if it's just for one sample, it's okay. But if you want to do it for a patient study or a lot of people that you want to monitor, it takes a long time to put it on there, to take off the layer that is on the lymphoprep. So a lot of handling. So we decided, okay, let's work with whole blood. And because the Comet assay has been applied to it successfully, I mean, the DNA damage version, we said, okay, we should do it for DNA repair as well. So we did aliquots of 250, which is the aliquot that I say is the most ideal. But we also went up higher because we need quite a lot of cells to extract the proteins. And then you can see if we do this, so one aliquot, or if we combine two aliquots, or a full 500 aliquot frozen, and then we wash it 2 times with PBS after defrosting. You will think, yeah, but the cells will die. Actually, they don't. So the red blood cells burst, but the white blood cells are still intact. You lose some, but still you get a good palette. And then we also isolated the lymphocytes in parallel to compare. And actually, you can see that we got some quite nice results. So here, probably we got too few cells or something happened in the process. But here with this aliquot of whole blood, we managed to get similar data to the isolated PBMCs. And yeah, that's promising. So it's very preliminary data. We're going to optimize this further. So hopefully in the future, that will speed up the processing of the blood for the repair assay. Also, there has been an optimization of the repair assay for the use of Drosophila. So that opens up also new applications that you can use as sentinel animals for exposure. And as Amaya showed you yesterday, these have all been applied to the Comet assay. So why not apply it also to the DNA repair assay? You just have to think about, okay, what do I want? Do I want to use the whole animal, like with the Drosophila, or do I want to isolate parts of these animals? The most important thing, I'm almost there. So the most important thing when you decide to use a sentinel animal for environmental biomonitoring is that you optimize the assay for it. So one of the steps is the extraction, right? Remember, and then you might have noticed this checkpoint. That's how we called it in the Nature protocol paper. The most important thing is that you optimize the protein concentration you use, because otherwise you get this, what we call non-specific activity, because you get the same signal for your non-exposed substrates and your exposed substrates. So there's some non-specific cutting going on there. So then you have to find the best concentration to work with. So for the brain tissues, in that study I did there, I used 1 milligram. For the liver, we didn't see that effect, but we decided to go with 5. So you always look for the biggest difference between the two. And there's another thing. So here you might have noticed that I said based on protein concentration. This is what we've been doing so far, and this is what has been explained in the Nature protocol. Maybe in a few years we will publish an update on that, because what we've noticed is in a rat study, we wanted to compare different tissues. And we wanted to compare liver, lung, blood, adipose tissue, and muscles. And we came into troubles with the muscles. Because when we wanted to use the same concentration, so for liver, we used 3 milligrams per milliliter. And we wanted to do the same for muscles, we didn't see anything. So we went up. But we already had to go up to 10 or 20 milligrams per milliliter to see anything in the muscles. Like, come on, this is ridiculous, you know, like it's too high. And then if you do it for other tissues, you get non-specific activity. So it was like, what's going on? But imagine a muscle cell. It has a lot of different proteins, the myosines, for instance, like it's a lot of other proteins in there. So probably if we use the protein assay to determine the protein level, you get an overestimation, or how should I say, like the proportion of your repair enzymes in there is in ratio lower. Follow what I want to say? So yeah, then the amount of repair enzymes you put on for the muscles is probably lower than for the liver. So we said, okay, maybe we should change it and base it on DNA concentration. So what do we do now once the extract has been prepared? We use a fluorescent dye to actually assess how much DNA there is in the extract. And then we know that a cell has on average 6 picogram of DNA in it for the nuclei, and then we count it back to the number of nuclei. And then we use this to do a dilution. So here you see a dilution. Well, actually, this is already the final bare activity, so we already subtracted. This is the net effect. You can see that we use different numbers of nuclei, and that you see that there is also a difference seen there. So yeah, this is now obese and lean animals for the liver. But you see that we can pick up quite a nice repair activity there. We still have to see how it corresponds to each other, because if you see here, for instance, 1 milligram, and here is 10 times lower, there's something going on there still. Like I say, this is also preliminary work from a bachelor project, but it is promising. So maybe in the future, we don't use protein concentrations anymore, but we will use DNA concentrations. Yeah. Okay, so that brings me to the end. So what are the take-home messages? Like I said, study multiple biomarkers in parallel, because just looking at repair, it can either be that, yeah, it's triggered, or you're just healthy and you don't have to have a high repair at that moment. Take into account gene-environment interactions whenever you can. Optimize the repair assay, like I just said, for every new tissue or cell type that you want to use. And that can be done either on proteins. So if you just want to look at one tissue between groups, you can use the proteins. If you want to look between other tissues, use the DNA content. Take into account positive assay controls. I didn't really talk about it, but Amaya talked about the FPG yesterday. So what we also do is we have exposed cells that we incubate with FPG next to the extract, just to see if the assay worked. If we see a signal with the FPG, we know the incubation reaction worked. But if you want to read more about it, you can go to the Nature protocol paper. And yeah, like I say, start applying it in environmental biology. So if you think about any sentinel animals, like I think the assay is ready to be applied in other fields. So would be interesting to see. So I promised you a few QR codes. So this is the Comet cost action that a lot of us were involved in, but there you can also find links to the films and to papers. The other one is the ICAO-G, so the International Comet Assay Working Group. Goran is the chair of it. So if you want to know more, you can talk to Goran, or you can use this QR code to check out the website. Also there you find links to the films and papers, the Nature protocol papers as well.